Science, 23, Human Genome Project.
The Human Genome Project, Deciphering the Blueprint of Life.
Mapping the human genome, the Human Genome Project, completed in 2003, was an unprecedented scientific endeavor that involved mapping the entire human genome, unraveling the sequence of nucleotides that make up our DNA. This monumental achievement provided a comprehensive blueprint of the human genetic code, consisting of approximately 3 billion base pairs. The genome sequence serves as a reference for understanding the organization and structure of genes, non-coding regions, and regulatory elements within our DNA, paving the way for advancements in medicine, genetics, and biological research.
International collaboration.
The Human Genome Project represented a remarkable example of international collaboration, involving scientists, researchers, and institutions from around the world. This collaborative effort brought together expertise in genomics, molecular biology, computational biology, and other related fields to tackle the immense challenge of sequencing and interpreting the human genome. The project fostered the sharing of data, resources, and knowledge across borders, transcending national boundaries and uniting scientists in a collective pursuit of understanding the genetic basis of human life. Gene identification. One of the major achievements of the Human Genome Project was the identification of thousands of human genes. Through comprehensive sequencing and computational analysis, researchers were able to annotate and catalog the genes present in the human genome. It is estimated that the human genome contains approximately 20,000 to 25,000 protein-coding genes, which encode the instructions for building the diverse proteins that govern biological processes in our bodies. Identifying these genes has provided a foundation for understanding the molecular basis of human health and disease. Variation in DNA. The Human Genome Project contributed significantly to our understanding of genetic variations and similarities between individuals. By comparing the genome sequences of multiple individuals, scientists discovered that humans share about 99.9% of their DNA, highlighting the fundamental unity of our species. However, the remaining 0.1% of genetic variation accounts for the diversity seen among individuals, including differences in susceptibility to diseases, response to medications, and physical traits. This knowledge has advanced our understanding of human diversity and the complex interplay between genetic and environmental factors in shaping individual characteristics. Genetic diseases. The completion of the Human Genome Project has been instrumental in elucidating the genetic basis of numerous diseases. By correlating genetic variations with disease phenotypes, researchers have identified specific genes associated with inherited disorders, such as cystic fibrosis, Huntington's disease, and sickle cell anemia. This knowledge has revolutionized diagnostic approaches, enabling genetic testing for early disease detection and personalized treatment strategies. The understanding gained from the project has paved the way for targeted therapies, gene editing techniques, and gene replacement strategies, offering hope for individuals affected by genetic disorders. Bioinformatics. The ambitious scope of the Human Genome Project necessitated the development of new computational tools and approaches, giving rise to the field of bioinformatics. Bioinformatics combines biology, computer science, and statistics to manage, analyze, and interpret large-scale biological data, including genome sequences. The project spurred the development of sophisticated algorithms, databases, and software tools for storing, analyzing, and visualizing genomic information. Bioinformatics has become a vital discipline in genomics research, facilitating the exploration of genetic data, the identification of genes and regulatory elements, and the understanding of complex biological processes. Publicly accessible data. The data generated by the Human Genome Project, including the genome sequence and associated annotations, have been made freely available to the scientific community and the public. This open-access approach to genomic data has catalyzed research in diverse fields, allowing scientists worldwide to explore and build upon the foundational knowledge provided by the project. The availability of this data has accelerated discoveries, fostered collaboration, and empowered researchers to investigate the genetic basis of diseases, develop new therapies, and gain insights into human evolution and population genetics. Ethical, legal, and social implications. The Human Genome Project raised important ethical, legal, and social implications that extend beyond the realm of science. The project prompted discussions and research on issues such as privacy concerns, genetic discrimination, consent for genetic testing, and the responsible use of genetic information. It led to the establishment of ethical guidelines, policies, and regulations to ensure the ethical conduct of genetic research and protect individuals' rights. The project highlighted the need for ongoing dialogue and interdisciplinary collaboration to address the complex ethical challenges arising from the rapid advancements in genomic science and technology. Technological advancements. The Human Genome Project stimulated significant advancements in DNA sequencing technologies,
revolutionizing the field of genomics.
The project served as a catalyst for the development of faster, more accurate, and cost-effective sequencing methods. Sequencing technologies have evolved from the laborious and time-consuming Sanger sequencing method used during the project to high-throughput next-generation sequencing platforms that can rapidly sequence entire genomes. These technological advancements have democratized genomics research, allowing scientists to explore the genetic underpinnings of diseases, unravel complex traits, and advance personalized medicine approaches.
Cancer research.
The comprehensive map of the human genome generated by the project has had a profound impact on cancer research. By comparing the genome sequences of cancer cells to normal cells, researchers have identified genetic mutations and alterations associated with different types of cancer. This knowledge has led to the discovery of oncogenes, tumor suppressor genes, and other genomic aberrations driving cancer development and progression. The understanding gained from the project has paved the way for precision medicine approaches, including targeted therapies and immunotherapies, which aim to treat cancer based on the specific genetic characteristics of individual tumors.
Pharmacogenomics.
The insights provided by the Human Genome Project have facilitated the field of pharmacogenomics, which examines how an individual's genetic makeup influences their response to drugs. By studying genetic variations in drug metabolism enzymes, drug transporters, and drug targets, researchers can tailor medication choices and dosages to individual patients, optimizing treatment efficacy and minimizing adverse drug reactions. Pharmacogenomics has the potential to revolutionize healthcare by enabling personalized medicine approaches that consider an individual's genetic profile when prescribing medications.
Understanding evolution.
The Human Genome Project has contributed to our understanding of human evolution, migration patterns, and our shared ancestry with other species. By comparing the human genome to the genomes of other organisms, including closely related primates, researchers have identified genetic changes that occurred during the course of evolution. This comparative genomics approach has shed light on the genetic adaptations that have shaped our species and provided insights into the evolutionary history of human populations across different regions of the world.
Epigenetics.
The Human Genome Project laid the groundwork for the study of epigenetics, which investigates changes in gene expression that occur without altering the DNA sequence. Epigenetic modifications, such as DNA methylation and histone modifications, play a critical role in regulating gene activity and can be influenced by environmental factors. The project's findings, combined with subsequent research, have highlighted the significance of epigenetic mechanisms in development, disease susceptibility, and response to environmental cues. Understanding epigenetic modifications has opened new avenues of research for exploring the interplay between genetics, environment, and complex traits. Functional genomics. Following the completion of the Human Genome Project, scientists embarked on understanding the functions of genes and their interactions within biological systems, leading to the emergence of the field of functional genomics. Functional genomics aims to decipher the roles and relationships of genes, proteins, and other molecular components in the context of cellular processes, tissues, and organisms. Through techniques such as transcriptomics, proteomics, and metabolomics, researchers have gained insights into gene expression patterns, protein functions, and molecular networks, advancing our understanding of biological systems and disease mechanisms.
The human proteome. The Human Genome Project laid the groundwork for exploring the human proteome, the complete set of proteins encoded by the genome.
Understanding the human proteome is essential as proteins are the workhorses of cells, responsible for carrying out crucial biological functions. Building upon the genomic information provided by the project, researchers have embarked on large-scale proteomics initiatives, aiming to catalog and characterize the full complement of proteins in different cell types, tissues, and disease states. This knowledge provides a deeper understanding of protein function, cellular processes, and the development of novel therapeutic targets.
Genetic testing.
The Human Genome Project has paved the way for the development and widespread availability of genetic testing for inherited disorders. Genetic testing allows individuals to determine their risk of developing certain diseases or to identify carrier status for genetic conditions. By examining specific genes or variations associated with disease, genetic testing provides valuable information for disease prevention, diagnosis, and treatment. The project's findings have empowered individuals and healthcare professionals with genetic insights, enabling informed decision-making and personalized healthcare approaches. Informed consent and privacy. The Human Genome Project sparked discussions and raised concerns about issues related to informed consent, privacy, and the potential for genetic discrimination. The project's implications necessitated careful consideration of the ethical and legal frameworks surrounding the use of genetic information. Safeguarding individual privacy, ensuring informed consent for genetic testing and research, and protecting against discrimination based on genetic information became focal points of legislation, policies, and guidelines.
These considerations continue to shape the ethical practices and regulations governing the use and dissemination of genomic data. The human microbiome.
Understanding our own genome has led to increased interest in the genomes of the microbiome, the trillions of microbes that inhabit our bodies. The Human Genome Project indirectly influenced the study of the human microbiome by revealing the interconnectedness between human genetics, the immune system, and the microbial communities that live within us. The project's findings spurred research into the role of the microbiome in health and disease, shedding light on its impact on digestion, metabolism, immunity, and various physiological processes. The study of the human microbiome represents a significant extension of the project's impact on understanding human biology. Genetic ancestry testing. The completion of the Human Genome Project paved the way for the rise of genetic ancestry testing, which allows individuals to trace their genetic heritage and learn about their ancestral origins. By comparing an individual's genome to reference populations, these tests provide insights into geographic ancestry, migration patterns, and genetic connections to different populations around the world. Genetic ancestry testing has become increasingly accessible to the public, fostering a deeper understanding of human population history. Gene therapy. The comprehensive mapping of the human genome by the project has provided a foundation for potential treatments involving the alteration or replacement of faulty genes. Gene therapy aims to correct genetic defects by introducing therapeutic genes or modifying existing genes in a patient's cells. The knowledge gained from the Human Genome Project has enabled researchers to identify gene mutations underlying genetic disorders and develop targeted gene therapies to address these conditions. While gene therapy is still an evolving field, the project's insights have paved the way for innovative approaches to treating inherited diseases. Model organism genomes. As part of the Human Genome Project, several model organism genomes were sequenced, providing invaluable tools for biomedical research. Model organisms, such as mice, fruit flies, and roundworms, share many genetic similarities with humans and serve as valuable systems for studying fundamental biological processes and disease mechanisms. The project's focus on sequencing these organisms' genomes has allowed researchers to explore the conservation of genes, pathways, and biological functions across species, providing insights that can be extrapolated to human biology. The availability of model organism genomes has accelerated discoveries, experimental research, and the development of new therapies in biomedical sciences. Thanks for watching. Please like, share and subscribe, and comment below if you agree, or would like to add your thoughts.